Good. So thank you so much for having me. My name is Manisha Munasinghe, and I'm a postdoc in Yaniv Bronvain and Nathan Springer's labs at the University of Minnesota. And I actually consider myself a theoretical population geneticist and an evolutionary geneticist interested in speciation, reproductive isolation, and genetic conflict. And while I don't have a direct link to a paper to share today, I'd love to just shout out that the work that I'm going to be talking about in a second is going to be on bioRxiv any day now, even possibly later today. And so if you're interested in some of the stuff that I talk about today, you can find more details there. So today as I mentioned, I am primarily interested in how inter and intragenomic conflict may generate hybrid incompatibilities that contribute to reproductive isolation. And as part of my work in that area, I've been thinking a lot about transposable elements. And so today I'm going to be sharing some fun insights from some population genetic simulations we've been building and exploring that look at the way insertion preferences influence the long-term invasion trajectories for transposable elements. I'm going to start by giving just like a little bit of background to motivate and frame our conversation before transitioning into our model design and our results. And finally, because this is a group of folks interested in maize genetics, I am going to end by just comparing some of what we're seeing in terms of TE copy number evolution with what we're seeing in maize and how our thinking of TE evolution has grown as a result. So transposable elements, or TEs, are mobile repetitive DNA sequences that actively increase their copy number and propagate themselves within genomes. TEs can be divided into classes, subclasses, superfamilies, and families, depending on their replicative mechanisms, ancestral origins, and sequence similarity, which highlights the genetic and mechanistic diversity of TEs. TEs have been highly successful at invading eukaryotic genomes, but there is significant variation between species as to the proportion of their genome occupied by TEs. And it is unclear what factors determine not only how much of the genome is occupied by TEs, but also how that final proportion is distributed between the present TE families. Some TE families contribute relatively little to this overall proportion, with only a handful to tens of copies present in a genome, while others contribute tens of thousands of copies. And this level of variation extends between species and is present even within a single species, where there can be meaningful differences in the mean copy number of a TE family or superfamily within different lines or populations. And here I'm just showing an example of this in maize. Now, high TE abundance is unusual given the expectation that novel TE insertions are likely to be deleterious, as they may insert into functional genes, alter heterochromatin formation and gene expression patterns, and induce large structural changes via ectopic recombination. So this raises a question of how or why certain TE families are capable of reaching high copy number within the genome. So in the 1980s, several theoretical population geneticists, notably Charlesworth and Langley, developed a series of deterministic population genetic models to explore how the mean copy number per individual for a given TE family could change over time, given their potentially deleterious effects. These studies demonstrated that the mean copy number could increase and eventually stabilize at an equilibrium point, depending on the extent of transposition and excision for the TE family, the effective population size of the host population, and the strength of selection against high TE copy numbers. Essentially, an equilibrium is reached when either all occupiable sites are filled with TEs, or the transposition rate is scaled down such that the number of new TEs each generation matches the number lost to drift, selection, and excision. Now, these models all make several notable assumptions that are well-founded and grounded in standard PopGen modeling and TE biology, but are slightly limited in their accuracy. So for example, these models assume a constant or infinite population size, which is standard practice. But you can imagine that as the mean copy number of a TE family grows, so does the associated deleterious load as you obtain more and more novel insertions at potentially deleterious sites, which could affect individual fitness, resulting in reductions to population size over time, and even putting the population at risk of extinction. These models also assume an inverse relationship between TE copy number and transposition probability, essentially incorporating in a simplified mechanism for either autoregulation or host repression, which is once again well-founded, but how and when these processes evolve and act to limit transposition isn't fully understood yet. Right? Just because the host genome is capable of silencing TEs doesn't necessarily mean that it's evolved to recognize and silence a novel TE when it initially invades. So there may be periods of uncontrolled transposition during TE proliferation. And finally, for the most part, these models assume either a fixed effect, where each new insertion has the same selective consequence, or an increasing effect, where the selective effect of a new insertion is slightly larger than the effect of the previous insertion. But we know that this isn't quite true. So TE families tend to preferentially insert into specific DNA sequences or features within a genome. So L1 or allo elements, for example, are enriched in opposite DNA isochores, while other TEs like Het A or Ti1 preferentially insert into telomeric or subtelomeric regions. TE families that preferentially insert into heterochromatic or intronic regions may result in new insertions that are selectively neutral, as they avoid the deleterious effects associated with inserting into a gene. Consequently, insertion preference may not only be the result of structural differences between TEs, but also an evolved trait that impacts the selective effect of new insertions. And so insertion preference may therefore represent not just the nucleotide sequence or feature a TE family inserts into, but it may also dictate an underlying distribution of fitness effects for each new insertion. And it's been hypothesized that TE families could evolve a preference for neutral insertion sites so that they can grow their copy number while also minimizing the effects of new insertions by sort of minimizing their cumulative deleterious load. So how often TEs transpose and the distribution of fitness effects for insertions are expected to determine how the mean copy number of a TE family changes over time. But despite several hypotheses about how these processes influence copy number, we haven't really tested this. So it's obvious that unconstrained transposition would cannibalize the host genome and ultimately drive the population extinct. But we know comparatively little about how variation in TE biology impacts this process. If a TE family is not initially lost due to either genetic drift or selection, how does its mean copy number change over time? And is it possible to maintain high TE copy numbers in this model without driving the population extinct? So how rapidly does that copy number change? And how is that correlated with population size and whether or not those increases in copy number drive populations extinct? And so ultimately, we were really curious about how these two key parameters, transposition probability and insertion preference, could potentially act together to sort of reach high copy numbers without decreasing population size or contributing to extinction. So to essentially dig into this question, we set out to use SLIM3, a flexible forward in time genetic simulation framework to better understand how these aspects of TE biology influence the mean copy number of the TE family and host population size over time. The awesome thing about SLIM is that it is a stochastic simulation framework, which differs from previous deterministic models. And it also has a ton of flexibility. So we can sort of build a specific genomic architecture. We can actually set the sort of distribution of fitness effects for new insertions, and we can model transposition and other aspects of TE biology. So we started by just considering a single diploid hermaphroditic population that reproduces sexually with discrete generations. We initialized the population with 1,000 individuals and use this as a hard carrying capacity throughout our model. We do allow the population size to fluctuate depending on the fitness of individuals, which we need if we want to explore when extinction occurs. We employ viability selection such that the fitness of an individual determines the probability that they survive to the next generation to serve as a potential parent. It's worth mentioning that we don't consider any form of fertility selection. We only consider viability selection here. So if an individual survives this viability selection, that offspring can then serve as a potential parent, but parents are selected randomly to generate offspring. We consider a 5-chromosome model for our sort of genomic architecture, where each chromosome contains 100,000 distinct sites with a uniform recombination rate of 1 × 10⁻⁵. So there's sort of higher levels of recombination in this model. To model insertion preference, we actually treat it as the chance that a novel insertion carries either a neutral or deleterious selective effect. For simplicity, we pre-assign each site in the genome to one of 4 selective classes that represents the fitness effect to an individual if a TE inserts into that site. So either neutral, mildly deleterious, modestly deleterious, and massively deleterious. The proportion of each class in the genome is determined by the parameter newt_p, which represents the probability that a site in the genome is neutral. So functionally, high neut p-values means we expect the TE to result in more neutral novel insertions. When actually determining how these sites are distributed, we distribute them randomly. So there's no artificial clustering of deleterious sites together. But we sort of essentially determine the number of neutral sites and then evenly divide the remaining sites between these 3 deleterious classes. So once we have this genomic architecture, we then have to actually introduce the TE. So in the first generation, we introduce a single neutral TE in the genome of a single individual, analogous to a horizontal transfer event. Transposition is modeled as a Poisson process, with the parameter TE jump P representing the probability that a single copy of a TE will copy and jump elsewhere in the genome. So the more copies of the TE you have in the genome, the more expected jumps you'll have in the next generation. The site for each new insertion is randomly chosen, and the fitness effect of that novel insertion is then determined based on the assigned fitness consequence for a TE that inserts into that site, as determined by that neut P parameter. So it's worth mentioning again that TE jump P is fixed for the entirety of a simulation, meaning we do not rescale or limit transposition. So for our purposes, we're sort of introducing a TE, and we will, and what I'll refer to in the rest of this talk, we refer to that TE as belonging to a specific TE family. And here TE family refers to a unique parameter combination of neut P and TE jump P. So insertion preference and transposition probability. So we introduce this single TE that belongs to this family, is then allowed to proliferate, and we sort of track its copy number in the population every generation over time. So we allow this to go until one of 3 endpoints are reached. So the first thing that can happen is that we actually lose the TE. It's possible that all copies of the TE family could be lost from the population due to either genetic drift or selection. The next thing, if we are capable of retaining the TE in the population, I say we, the simulation, if the simulation retains the TE, the next thing that can happen is the population size can hit 0. That means that no individual has essentially survived after viability selection and the population crashed. And we record that outcome as resulting in population extinction. And so finally, if neither TE loss or population extinction occurs, the only remaining endpoint is that both the TE family and the population persist for 50,000 generations, which is our generational time limit. So we introduce this TE and we essentially track its proliferation until we see how often these outcomes occur. And we sort of continue that process of introducing TEs and documenting what's happening until we either have 1 million TE loss events, which we need because due to drift expectations, we do expect that the TE is going to get lost very frequently. Or we have 100 combined outcomes of population extinction or dual survival. And so what that allows us to do is that both for a specific parameter combination and across parameter combinations, we can look to see how often these outcomes occur. So in what regions of our parameter space do we see either an excess or a lack of TE loss events? Are there regions of our parameter space where we more often see extinction versus dual survival? And so we sort of use that as that's what we're really looking at here, these outcomes. Okay, so with that, I'm going to go ahead and jump into some of our results. So like I said, by far the predominant outcome across all of our parameter space is TE loss, which is not unexpected given the very low initial allele frequency. So for the next couple of slides, I'm going to show you some heat maps that have a very similar structure. So I just want to walk through what we're looking at. So here we have a heat map where the rows and the columns represent those the unique parameter combinations we're interested in. The rows of this heat map are our transposition probability with highly proliferative TEs on the top. Our columns are neutral insertion preference. So essentially, how often does that insertion result in a neutral insertion site? And so things that preferentially insert into neutral sites are going to be on the right of this heat map. Right now, each cell is going to be colored by the number of TE loss outcomes, with darker colors indicating more TE loss. So across all replicates for a given parameter combination, we find that at least 85% of all replicates result in TE loss, with over 68% and 95% of all TE loss outcomes occurring within 10 and 50 generations respectively. So we believe that this is primarily due to genetic drift, as the initial TE is neutral and begins with frequency 1/2000. Now, neutral mutations, not TEs, fix with a probability equivalent to their allele frequency. And we find that for the most part, the number of TE losses aligns with drift expectations across our parameter space. And we can sort of look to see where deviations from this expectation occur. And we see that we actually have a reduced number of TE loss outcomes at the top of our parameter space, the top of this heat map. So we want to be able to look and say, okay, if we're not having TE loss, we're either having population extinction and dual survival. So where are we seeing those outcomes occurring? So once again, same sort of similarly structured heat map, but now each cell is going to have two different triangles. The upper right triangle shows the number of population extinction events, while the bottom left triangle shows the number of dual survival outcomes. Once again, with both the intensity of the color indicating how often that occurred, with the sort of maximum being 100 of those outcomes. So when the transposition probability is low, we observe the highest density of dual survival outcomes. In fact, the only time we ever exclusively observe dual survival is for our lowest transposition probability. As we increase the transposition probability, a boundary between dual survival and population extinction begins to emerge. That starts when the preference for neutral insertions is high. And as we sort of continue to increase the transposition probability, we observe more and more population extinction outcomes that extends into regions where the preference for neutral insertion sites is lower. And then finally, once transposition gets sufficiently high, population extinction takes over. And we find that actually the proportion of population extinction outcomes relative to TE loss also increases in this space. So not only are we no longer observing dual survival events, but we're also finding that these population extinction outcomes occur more frequently. So we have this sort of like high extinction at the top of our space. But what we found to be really curious is that we actually found that a preference for neutral insertion sites in our model was more likely to result in population extinction, which is sort of counter to what we initially hypothesized. So at this point, we wanted to dig in and sort of think, okay, what's going on in these population extinction outcomes? So to examine the factors associated with population extinction, we examined the timing of extinction events and the copy number of neutral and deleterious TEs in the generation preceding extinction in our 5-chromosome model. Sorry, one sec. So what we found was that populations with higher transposition probabilities go extinct more rapidly across all insertion preferences. So here what I'm showing you transposition probability in each cell with high transposition probabilities on the right, and our neutral insertion preference is on the x-axis of each of these cells with high neutral insertion preferences on the right. And sort of the generation that population went extinct is on the y-axis. So things that are smaller actually mean that population went extinct more rapidly. So we find there's this really dramatic decrease in the time to extinction as we increase the transposition probability, with things at that highest space going extinct in less than 500 generations for the most part. If we fix the transposition probability and just maybe look at one cell, we find that lower neutral insertion preferences, so more deleterious insertion sites, actually delayed the time to host extinction in comparison to higher neutral insertion preferences. But it's worth mentioning that the time to extinction eventually stabilizes across these higher range of neutral insertion preferences. So once again, higher transposition probabilities dramatically reduce time to population extinction. But we still see this kind of strange result again of, well, preferentially inserting into neutral insertion sites, not only are we seeing population extinction more frequently, they're also going extinct more rapidly. So we then decided to look at the relationship between the final mean copy number of neutral and deleterious TEs and extinction events. And so here we found that there's actually significant variability in the final copy number of the TE family in population extinction outcomes, with it ranging from around 20 copies per individual to around 270,000 copies in our most extreme cases. And what we found is that the final copy number tends to decrease as we increase the transposition probability and reduce the preference for neutral insertion sites. So you might have initially thought, well, if you have a higher transposition probability, you're the most proliferative TE, you'll actually reach the highest copy number. And we didn't find that to be true. Instead, we actually found that our highest copy numbers for our neutral TEs, which are really driving the total copy number, are really observed under moderate transposition probabilities and high neutral insertion preferences. So once again, we have this sort of counterintuitive result where transposition is actually, higher transposition is actually not helping you reach those high copy numbers. So when we looked at the deleterious copy number, we found that there is substantially less variability in the final copy number here, which actually makes sense to us. We expect that there is some sort of maximal deleterious load these populations can withstand. And what we're observing in this final generation before extinction are these populations essentially converging on this point. And so they're all one generation away from reaching this variability. And the difference in sort of how close they are to this point really depends on that transposition probability, right? If you have a higher transposition probability, you need fewer copies to generate enough insertions to get you to that point. So what we then decided to do is we said, okay, let's just for a second, let's fix transposition probability and look at how the deleterious copy number changes over time as it relates to the neutral copy number. So here what I'm showing you this is a fixed transposition probability for our moderate transposition probability with sort of 5 distinct neutral insertion preferences with high preferences for neutral insertion sites on the right. The x-axis is sort of the generation and the y-axis is the deleterious copy number. And so for each replicate, we're plotting that change in deleterious copy number and we're coloring the line by the neutral copy number at that time. And so what you can see is that the deleterious copy number, and I think this is most clearly demonstrated in the sort of more high insertion preferences, the deleterious copy number actually stays low. It's essentially at like close to 0 until there's sort of a critical threshold of TEs, neutral TEs, that is reached at which point the deleterious copy number grows exponentially. And we find that this increase in deleterious TEs is perfectly correlated with population fitness, which is expected. And what's happening is the population is essentially entering into an extinction vortex. So there's this sort of triggered and unescapable increase in deleterious TEs that is highly dependent on the neutral copy number. And so while there may be variation between replicates as to when that point is reached, once it is reached, there's incredible consistency in our stochastic simulations as to the copy number and fitness trajectories. And you're going to sort of see this by the coloration of these lines. When a trajectory or a replicate actually increases may vary, but the coloring and timing of that trajectory is incredibly consistent. So what do we think is happening here? So what we started thinking about as well, we know that selection can remove deleterious TEs. So as long as they can remove deleterious TEs faster than they're added, the population is safe. So during that initial invasion, when the TE copy number is low, there's only a handful of TEs and only a small proportion of those are expected to be deleterious. And so selection is actually really capable of removing those deleterious TEs and keeping that copy number low, which is why we see for a while in those trajectories sort of a delayed increase in that copy number. But what's crucial here is that selection does not act on neutral TEs. So their copy number can actually increase. And so the more you preferentially insert into neutral TEs, you can actually grow your copy number faster. But we have 2 key parameters here, right? We have transposition probability, which determines the number of new jumps each generation. And so the more TEs you have, the more new jumps you're going to have. And then we have this insertion preference, which essentially determines how many new insertions are neutral versus deleterious. And that is actually going to stay constant across generational time. The proportion should be the same, but you might be having more and more TEs. So the more TEs you have, the more deleterious TEs you're also going to get. So essentially what we think is happening is that there's some sort of crucial threshold. We're given this sort of neutral copy number, you are always going to get more deleterious TEs each generation than you can remove via selection. And it's at that point that the population is headed for extinction. So our results actually contradict this hypothesis that preferentially inserting into neutral insertion sites actually protects the population. What it's doing is it does actually allow the copy number to grow faster, to grow more rapidly, which we sort of see with those transposition, the neutral copy number graphs. But the sort of downside to that is they also churn out that result in more deleterious insertions. So there's this like very cool relationship between transposition probability and insertion preference as it relates to copy number and then extinction. So before I go on and talk about some dual survival outcomes, I just want to mention that we did consider a couple of different genomic architectures and TE biologies in our models. So we did consider a sort of one model low recombination rate model. So with a lower recombination rate, and what we found is that our results didn't change. We just actually had more instances of TE loss. Essentially, we had early linked selection against deleterious TEs that were strongly linked to neutral TEs. So when we found that in regions of lower recombination, it was actually harder for TEs to invade because there was intense selection against those deleterious TEs, and those were linked very often to our neutral TEs. And so our neutral TEs were lost as a result. We also considered two additional aspects of TE biology: random excision and the inclusion of non-autonomous elements. So random excision occurs when a TE copy is sort of randomly lost, so that you actually expect a decrease in the copy number. And what we found, once again, is that it didn't actually change any of the general trends we were seeing. We still saw these same sort of results, but we just saw a higher proportion of TE loss outcomes, which once again kind of makes sense because you're more likely to lose that TE if it randomly excises out. And then the last one we considered is the inclusion of non-autonomous elements, essentially elements that could impact fitness but couldn't actually contribute to any new insertions. And we found, once again, didn't change any of the trends we were seeing as they relate to population extinction, the trajectories, the proportions, but we saw that it did increase the amount of TE loss. So none of those things that we incorporated could actually rescue this phenomenon. We found that this was actually quite consistent in the face of uncontrolled TE transposition. A preference for neutral insertion sites is not sufficient to actually protect the population in a meaningful way and allow the copy number to be high without resulting in population extinction. Sorry. Okay. So we have this really sort of counterintuitive but cool result in population extinction, but we also did want to look at what's going on with dual survival. So this occurs less frequently in our model, but what we find is that if we look at the mean final TE copy number for observed dual survival outcomes, we find that for the most part, our dual survival outcomes have a much lower final copy number compared to population extinction. So for our dual survival outcomes, across all of our dual survival outcomes, just over 50% of all observed dual survival outcomes have a mean final copy number of less than 10. And just over 80% have a copy number of less than 100. Also, when we look at the range of copy number values between dual survival and population extinction, we find that the dual survival range is much more limited in comparison to the population extinction one. So the median final copy number for dual survival outcomes is about 9 copies. If we compare that to population extinction, we find that the median copy number for population extinction is actually 587 copies. Now, that isn't to say that we don't observe copy numbers greater than 100 or even 1,000 in dual survival outcomes. And you can sort of see that in this figure that I'm showing you here, which actually just shows, like, for our dual survival outcomes, how many of those final copy numbers fell into these different copy number bins. And when we looked at those trajectories for any copy number that was really above 1,000 in dual survival outcomes, we found that those fitness trajectories looked very similar to the fitness trajectories I showed you for population extinction events, which essentially suggested to us that those were populations that were actively declining and they were actually decreasing in size, but they just hadn't reached 0 by that generational time limit. Remember, we have a 50,000 limit. And so those are things that were declining but hadn't hit 0 by that 50,000 points, and so therefore were marked as dual survival. And so in general, we found it really, really difficult to find any sort of dual survival outcomes that resulted in high copy numbers that seemed stable, seemed that they weren't headed towards extinction. And so if we actually looked at, for our dual survival outcomes, it's like, what is the allele frequency of each TE? We found that when we sort of bend this across our parameter space, that almost always our dual survival outcomes have this sort of bimodal distribution, which once again kind of makes sense to us. Dual survival occurs as long as a single TE fixes in the population and the copy number, the mean copy number in the population, stays below some critical threshold. So we see this observation of sort of some number and even many numbers of high fixed TEs, and then some sort of like high frequency, very low frequency polymorphic insertions. But in general, we just didn't see high copy numbers in dual survival outcomes that we really felt were stable dual survival outcomes. So our simulation results showed that if a TE invades a naive population and is able to resist initially getting lost from that population extinction actually more often occurs, not only when the transposition probability is high, but also when that TE belongs to a family that preferentially inserts into neutral sites. So like I said before, there's this really cool relationship between if you have if you have high copy numbers, you can reach that if you preferentially insert into neutral sites, but it's almost like a ticking time bomb. You're getting higher and higher copy numbers, but there is going to be an explosion of deleterious TEs at a certain point that selection just can't keep up with. And at that point, you enter into an extinction vortex you can't recover from. We also find, we find that you can't, like I was saying, high TE copy numbers almost always result in population extinction. And while we do occasionally see those for dual survival outcomes, when we actually look at those fitness trajectories, we really think that those are populations that are going to go extinct. So we then really started to think about how then can we actually achieve high copy numbers in populations? And to us, this indicated that there's an absolutely crucial role for transposition regulation, which we didn't include in our model. And so for us, we wanted to see, is a neutral insertion preference sufficient to protect a population? And it's not, it's actually worse. So that really, really means that transposition regulation has to be key in achieving high copy numbers. And so mechanisms that modulate transposition are varied and well documented across a wide variety of TE families and among different species. And these mechanisms can broadly be categorized as either autoregulation or host repression. So autoregulation occurs when TEs evolve self-regulatory mechanisms to modulate transposition on their own copy number. And examples of this have been well characterized in both Drosophila and yeast, where a TE lineage could limit its own transposition dependent on its copy number. So as it gets a higher copy number, it actually silences its own proliferation. Host repression, on the other hand, occurs when the host genome evolves to identify TEs and keep them from transposing. And I've highlighted just a few of these mechanisms here. Chromatin modifications, germline silencing of TEs, post-transcriptional silencing of TEs. These are all examples of ways with which host populations evolve to silence TE proliferation. But it's worth mentioning that it is unclear whether all TE families are capable of monitoring their genome-wide copy number and subsequently reducing transposition, or whether that's an evolved trait given the right blend of biology and selective pressure. And sort of similarly, while many species have already evolved mechanisms for repressing TE proliferation, they may not have evolved the ability to recognize novel TEs. And so there's this sort of potential as well that host repression may also evolve in a manner that's dependent on the copy number of the TE family. So we've been thinking a lot about what are the sort of selective pressures influencing the evolution of transposition regulation? When and how do these mechanisms act to limit transposition? And so if you think about sort of as you're declining, there may be even more intense selective pressure for host repression to evolve. And if it does evolve, it may actually fix very rapidly. But that could also be dependent on copy number. So maybe you need to have a lot of copies so that the chance that you get two TEs right next to each other and form a hairpin loop that can then silence has to happen. You could also potentially have these sort of replicative bursts. So if you think about the pi-RNA system in Drosophila, you might have to land in a cluster, that TE, pi-RNA cluster then silences that TE, and that TE has to accumulate enough mutations that it's no longer recognizable by that silencing, and then it can take off again. And so these mechanisms that sort of modulate transposition could silence for a while and allow this sort of like increase in neutral TEs and purging of deleterious TEs. There may be several sort of cycles of transposition bursts and also population decline where you have this sort of interaction between these mechanisms. And so we're really, really interested in this area, and that's sort of where we're looking to next, this idea of the interaction between transposition regulation, the varied forms of transposition regulation, and how they sort of contribute to both the evolution of copy number, but also whether or not saving a population from extinction. And the last thing that I want to sort of end on here is this question of a sort of philosophical question maybe about TE copy number evolution. So when we started this project, we were thinking about how TEs could achieve high copy number without driving the population extinct. When I asked sort of frame those initial questions, I was like, well, is this enough to get to high copy numbers without driving extinction? And this stems from a really commonly held assumption that TE families are selfish genetic elements seeking to proliferate regardless of their effects on the host. And that might be true. But by doing this project, we were really confronted with the reality that achieving high copy number means very little, potentially, if it results in host population extinction, as the entire TE lineage is effectively wiped out from the population. So this really raises a question of how this selective pressure at the level of the population to sort of save the population from extinction competes with the selective desires of the TE family and the host population to replicate and reproduce. So has there been sort of a culling effect or even selection against on TE transposition to potentially limit transposition so you maybe have fewer jumps, but you maybe give yourself more time for the host to evolve silencing mechanisms or for you to modulate your own transposition? I'm not really sure about that, what exactly that is, but there is this question of, okay, is actually high copy number the expectation? And so just as like a very baseline first pass, we thought, okay, let's look at the copy number of TE families in a natural population, say some maze lines, to actually look at what that copy number family distribution actually looks like. So first, we leveraged the TE structural annotation of the maze B73 reference genome made by Michelle Spitzer and colleagues to look at the family size distribution in this line. And so what I did here is we just stratified TE families by their class, order, and superfamily, and then binned the mean final copy number, not the mean, the final copy number for all the families in that class in a similar way to how we did that for our dual survival outcomes. And so we bin these across, and the number of families per each of these categories is above. But what you can see is that there's actually a lot that are low copy. So in this annotation, we found that 75% of observed TE families contained only a single copy. And 95% of those families showed less than 10 copies. 1.2% of all of those families had copy numbers greater than 100, and I think only 4 had copy numbers greater than 10,000. And those were all LTR elements. So for the most part, we actually don't see many high copy number families in B73. We then sort of took this same sort of strategy and applied it to the EDTA-TE annotations of the NAM lines. So sort of same sort of strategy, but here for space, I didn't sort of bin by superfamily or code. I just took all families in those lines. And what you do see is that there is sort of remarkable consistency across these lines, but that there's a lot of them are actually really low copy number. And so we find that 50% of observed TE families across the NAM lines contain only a single copy. And 73% of those show less than 10 copies. Once again, a very small percentage, 4.8% of families had copy numbers greater than 100. And so when we actually looked at this data, and I would say that I'm talking about maze here, but we have actually looked at some data. We pulled some data from Arabidopsis as well. Arabidopsis is very similar. There's only a handful of copies that are over 10,000, or over 10,000, I think, yeah. So this idea of really, really high copy number TEs doesn't really seem to hold when we look at some of the data. And so it does seem that there's only really a handful of TE families that have actually been capable of achieving these high copy numbers. And so we've really been wondering about whether this question of high TE copy numbers being the expectation, and whether many TE families exist at relatively low copy number, less than 100 copies in host genomes, and whether there's sort of selective pressures on those TE families or the host that potentially limit transposition or to really evolve mechanisms that are watching transposition. That once again, that really raises a question for me, and I think I have time because I'm going to end soon, so we can talk about some of these questions. But this really raises a question for me as to like how these systems evolve and how they actually gauge copy number and when and how they actually limit transposition. So with that, I know I'm early, but I did expect that there would be questions, and I can see that there are quite a few. And so I want to just acknowledge several folks in the Springer and Bronfein lab, all of whom have helped me sort of think through these models and these results. And of course, thank the NSF postdoctoral research fellowship in biology, which funded this work. And with that, I'm just going to go back to this key simulation slide, and I would be happy to take questions.
Okay, thank you very much, Manisha. So the first question is by Jin Yang. So would you like to ask your question yourself?
Hi, Manisha. It's a good talk. So actually, my question is back to your simulation. I saw there's a very many other questions. I think essentially very similar. Our concern is that this T mutation is supposed to be, well, can be considered as a functional mutation. Itself, actually, the T transposition itself is mutated. Because so essentially, I kind of disagree that the more T you have, then the more transposition you're going to have, because over time this T will degenerate. So therefore, and also under host control. So therefore, I think the jump P or the T jump P parameter supposed to be a function of time. Have you considered that in your initial simulation or not?
So we have, like I said, we didn't ever rescale transposition, specifically because we did look at this question of, okay, how and when does host repression or rescaling of that transposition rate occur? And partially it was because as we were looking at some of the literature, we found that there were these statements about uncontrolled transposition resulting in extinction, but no one had actually looked at it. And so we didn't consider that. I will say that we have done some preliminary results that have incorporated rescaling of this TE jump P parameter in order to see, okay, if we do incorporate like a very rudimentary form of auto-regulation, whether or not that's capable of saving the population. And we do find that it is capable of rescuing populations from host extinction, but there is obviously a consequence to the copy number. So we still have this question of how do you then get to these really, really high copy numbers in a population without resulting in extinction? If you think about sort of the degeneracy of those transposition rates, you could potentially have where you do have, okay, eventually there's no more transposition. At that point, when you have only what would functionally be like no more autonomous elements left, you only have non-autonomous elements. It's just a matter of time where TEs are either going to fix in the population via drift or get purged via drifter selection. So you're once again going to see these reductions in copy number. And so I think it's a very fair question. I will say that we debated a lot about this idea of uncontrolled transposition regulation. And the reason why we decided to stick with it is because we didn't actually see any work that was actually looking at those effects. But I think it's a very, very fair question. But I don't think that it sort of conflicts with this idea we have of, well, then how do you get to these high copy numbers without resulting in extinction? And I think that's sort of where we're walking away from this question being like, how is it possible then? And how long would it take? How many sort of managed replicative bursts do you have to do in order to reach those copy numbers and not result in extinction? Yeah, I hope that answers your question. I think it's a really, really fair question, though. I will say we debated it a lot.
Thanks. Yeah.
Okay, I had a question myself, but I don't know. Yeah, so my impression is like the more you had that you said basically the more you have TEs, the more deleterious T insertions can get. And I don't get this because I thought that maybe if you have many T copies on a big genome size occupied mostly by Ts, then any new insertions have a high probability to fail within an existing T and therefore to be neutral. So I actually don't get that.
Yeah, so I will say we didn't model nested insertions. So that wasn't something that we did in our model. But what we're saying is that even if you have a really, really high neutral insertion preference in our model, say you have 0.999, which is I think the highest that we did in our model, 0.999, we expect those insertions to be neutral. There's still a 0.001% chance that you can that you're going to result in a deleterious insertion. And so essentially your copy number has to be very, very high for the number of expected deleterious insertions, even if it is so small as 0.001, to essentially get to the point where you have enough deleterious TEs that you can't escape it. So even if you preferentially insert into neutral insertion sites, that doesn't mean that you exclusively insert into neutral insertion sites. In our model, I don't know if there's a TE family that exclusively does that. But in our models, we don't actually assume that there's any way that you exclusively result in neutral insertion sites. And so what we're saying is that in those models, you actually do see that the copy number increase is quite large. That's where we get those like moderate transposition and really high neutral insertion preferences. Those are where we see copy numbers of like 270,000, which you could argue is kind of unrealistic. I don't think we've seen any TE families that have reached those copy numbers. But in order to get to population extinction, you had to get copy numbers that high, such that the number of expected deleterious insertions given your insertion preference is still high enough that you can't escape this extinction vortex. Does that make sense?
Okay. And then I had a second question about, so then you're thinking So what would be some prediction that you could test on actual population data? Because if I understood correctly, you think that some population would be more likely to go extinct than others because of TE. So I mean, what kind of population data you could test this hypothesis?
Yeah, so that's a great question. And we've also been thinking about this a little bit. So one of the things that we sort of run into is that you have this sort of survivorship bias when you're looking at natural populations, where these are populations, both the host population and TE lineages, that have survived this potential extinction cycle. And so we have been wondering, whether there's any potential in looking at collections and looking at like populations that have actively declined and potentially gone extinct and looking at their TE copy number. But we do have some concerns about looking at that, given the other effects that are associated with declining population size, such as inbreeding depression, and sort of how do you uncouple whether that extinction was driven by TE proliferation versus other factors. We have thought about like, well, what about new invasions, like a TE that has just invaded a population? And one of our concerns there is, as we talked about, a lot of times with these new insertions, they very, very often result in complete TE loss due to genetic drift. So you need to find a population that has had a recent invasion, that has invaded but not gotten to a high enough copy number that the expectation that you might lose the TE family isn't there. Yeah, I have to say that I don't actually know a great way to test this extinction hypothesis. I would be really curious if anyone has any ideas about that. Because like I said, I don't know how to look at populations that have historically disappeared as a result of a TE invasion. And so that we don't have data for that. We are looking at some hybrids. We have thought a little bit about hybrid incompatibility. Now, to be fair, that's not necessarily a naive invasion. You can have like a much larger influx of a much larger TE copy number introduction via hybridization. Well, we have been thinking about, okay, well, those, you might have uncoupling of whatever silencing mechanism silences TEs in one population that's lost in the hybrid. And so a great example of this is like hybrid dysgenesis in Drosophila, where the silencing mechanism can be lost in hybrids and you can have TE proliferation. And oftentimes those hybrids show reduced fitness. So we've thought about looking at like reductions in fitness in hybrids over time, maybe a proxy for looking at some of this, but I'm not quite sure how to go about it from like a naive invasion.
Okay, thank you. Marcus, you had a question?
Yes, thank you for this really clear explanation of the simulations. I was wondering if you had a chance to think about coming up with a deterministic expression of where this threshold would be, like, because I mean, the simulations show that there is this threshold. Do you have a sort of deterministic way to say where that is?
Yeah, so Yaniv and I had thought about it, and we did take a crack at it initially, but things get really messy, particularly when you start to vary population size. That's why a lot of these deterministic models, when they do actually look at these trajectories, don't vary population size. And so we had a lot of difficulty with it. We do think that it does exist. And so if anyone is a really strong in differential equations and would like to talk about that, we would, but we do agree with you. We think that there probably is actually an analytical result, because there's incredible consistency in our stochastic simulations as to like when those sort of trajectories kick off. But we did not have a lot of success trying to model that deterministically.
And you cannot get it out of your sequence of different parameter combinations to sort of have an estimate where that would be? Like say, until when does it go extinct and when does it not anymore?
So yeah, the best guess that I think we could try, and it might be worth trying, is we could track the slope of that fitness trajectory line over time and essentially try to determine when we see an increase in that slope, and then see if we go back and look at when those increases start happening, if there's what the copy number is at that time. That's, I think, maybe the best way that we could get it from the simulations, is we'd have to go through and look at those changes in slope. We haven't done that, but that is an interesting idea.
Thank you.
Yeah, thanks.
So there was then a question by Arcadio, but I think it was maybe similar to a previous question. I don't know if you want to know more about what you were asking.
Yeah, thank you. No, it was basically the same thing that you asked and she already answered. It's pretty cool. Thank you.
Okay. And now it's merits. A question by merits.
I think Marcus kind of had the same question. It was the how many generations does it take to reach that critical threshold? And yeah, I think
that generational time point. There is a little bit of variability in there, and that is a result of drift, right? So there is a frequency of drift about how long it takes populations to get to that threshold, but it does sort of seem to cluster within a range of about 100 to 500 generations for all of them. Yeah.
Sarah?
Yeah, nice talk. That's exciting to see. So you kind of got into this. My question that I wrote down is, do you have any predictions about why your model, what you're missing in your model to account for high copy number TEs? And that's going to be TE silencing, right, mechanisms. But then I'm kind of curious because, I like to think about TEs as sometimes having some functional good consequences, right? And so I'm wondering if you've thought about, or if there's a way to model the fact that some TEs could do something good. And so is there some constraint where if a family is going to be able to do something good, then it has to have low copy number, because otherwise the only way to get to high copy number is to be silenced?
That's a great question. So I will say that for this model, the reason we didn't include adaptive, but we were thinking about it as a potential, partly for computational complexity and visualization of results. But if you think about an adaptive TE, there is this question of one, how often does a family generate an adaptive TE? We didn't have a good estimate for that. But if you think about, okay, you have a population that's declining, there's some number of deleterious TEs, and then you get an adaptive TE. That adaptive TE is likely going to sweep through the population. And while it's sweeping, there's going to be a big bump in fitness that may actually rescue the population for a moment from this extinction vortex. And so you may have this bump back up, but once that adaptive TE is fixed in the population, you enter back into this process because your copy number is still high. And so fitness then is going to decline again. So we think that there's possibly some sort of like cyclic fitness trajectory that might occur if you incorporate adaptive TEs. And it's really going to depend on how often those adaptive TEs are generated and whether or not it can rescue fitness. But even if it does rescue, there are unrelated to TE copy number evolution, there are consequences for going through declining population sizes, sort of effects of inbreeding, depression, loss of genetic variability. And so you might have this boost from an adaptive, which actually, as it sweeps, would reduce genetic diversity at that site. But you might have this boost in fitness that rescues you from extinction, but you're still experiencing these effects of cyclic population declines.
Sure.
And so it is something we thought about, but I wasn't really sure like how often would you expect these families, depending on their insertion preferences, to then generate adaptive TEs. But when we started to think about like what it would mean for our fitness trajectories, we didn't think that it would potentially rescue them from the effects of this. It would maybe just delay it.
That makes sense. Thank you so much.
Yeah, of course.
Okay, we have a last question, I think, by Shanghai.
Hi. So I guess it's similar to what the first question is. So when will because you didn't consider the TE silencing mechanisms in the simulations, but it's kind of answered by what you say later on. So the basic challenge now is that you're not sure about when will the mechanism being evolved, right?
So to give a little like backstory on this project and how we actually came to this. So we actually were originally interested in modeling host repression evolution. And so as we were setting up some of these models, we didn't have this question of, okay, well, what is our expectation if we didn't have host repression? And we couldn't find any sort of deterministic model or any sort of pop-gen models about decline, because it's really difficult to do declining population sizes about what would happen. And so we essentially took our model and we removed host repression to see what would happen. And that's when we started to see some of these really weird counterintuitive results about a neutral insertion preference, which is really counter to our expectations, where we thought that would be protective, and we were finding that it really wasn't. And so as we started digging in and we started digging into it more and more, we found that there was actually a lot going under the hood there, which is why we have this project that's actually without transposition regulation. But we do fully agree with everyone that whether or not a completely uncontrolled, freely replicating TE is biologically accurate, we're not quite sure. But we do think that there is probably likely periods of time where there is uncontrolled transposition. And so some of our models probably have implications for that, how that interacts with host repression and autoregulation. We're really interested in, that's really what we're really interested in digging into.
Okay, thank you.
Okay, thank you so much, Manisha. So is there any more questions? So Ruben had to run, so he said, great talk, Manisha, thanks for presenting here.
Of course.
Okay, so if there are no more questions, I think we should see you next week. We will see you next week. And thanks again, Manisha, for being here with us today.